Okay, so welcome back to this next video in which we are discussing heart failure and heart failure medications. Okay, right, so we're in the process of discussing the normal calcium signaling within cardiomyocytes that causes contraction of the cardiomyocytes. Okay, so we've discussed the calcium homeostasis within the cytoplasm of cardiomyocytes when they're at rest. Okay, and how they maintain a very low cytoplasmic calcium concentration of around 10 nanomolar. Okay, then what we've discussed is how an action potential propagating along the sarcolemma of cardiomyocytes is then going to induce a calcium spike, as it's called, within these cardiomyocytes, which is when the cytoplasmic calcium concentration rises from 10 nanomolar to 1 micromolar. Okay, and then what we've discussed is how it then is reduced back down. So you start with the inactivation of the L-type voltage-gated calcium channels and the type 2 ranitidine receptors, and then what's going to happen is the calcium is going to be removed from the cytoplasm. 75% of it's going to be removed by circuit 2A and therefore pumped back into the sarcoplasmic reticulum, okay, and the other 25% is going to be removed by NCX1. Okay, and then you'll re-establish this equilibrium where you've got a 10 nanomolar cytoplasmic calcium concentration, you've got a certain concentration of calcium in the sarcoplasmic reticulum. That will cause a leak of calcium from the sarcoplasmic reticulum lumen into the cytoplasm of the cell, and at 10 nanomolar calcium concentration, the movement of calcium back into the SR by the circuit 2A is going to exactly balance the amount that's leaking out. Okay, and that's important because that means that we're always going to end up with the same calcium concentration in the SR, because that's the only calcium concentration that can be at equilibrium with 10 nanomolar cytoplasmic concentration. Okay, because at 10 nanomolar you will have a certain amount of calcium coming in to the sarcoplasmic reticulum through circuit 2A. Okay, and the only way you can be at equilibrium is if the calcium concentration in the SR is such that the amount of leak that you get is the same as the amount that's coming in. Okay, so that always assures that this calcium concentration has to be the same, providing the circuit 2A activity is the same. The only point at which you can be at equilibrium is that certain sarcoplasmic reticulum calcium concentration, and that is such an important concept to understand. Okay, it's going to be important in just a moment when we move on to discuss how sympathetic stimulation of cardiomyocytes is going to increase the force of contraction. Okay, so just before we do move on to that, I just want to draw you a little graph, okay, which will show the calcium concentration as a function of time, okay, in cardiomyocytes. Okay, so it starts off at 10 nanomolar, then it will spike up when an action potential propagates along to 1 micromolar. It will then come down as you relax, then you'll have another action potential. Okay, again, relax, and it will go on and on. So these are the calcium spikes, basically, in the cardiomyocytes. So this is a calcium spike. Okay, right. So what I now want to discuss is how sympathetic stimulation of cardiomyocytes is going to increase the force of contraction of cardiomyocytes. Okay, and it does this by increasing the size of the calcium spike. So basically, once you've released adrenaline or noradrenaline onto the cardiomyocyte and stimulated beta-1 receptors, your calcium spikes will now look like this rather than like that. Okay, and having a larger calcium spike is then going to mean that you activate more cross-bridge cycling, and therefore the force of contraction that the cardiomyocyte can generate is going to be greater, basically. Okay, so larger calcium spike, more cross-bridge cycling, more activation of the sarcomeres, more force generated, and that's how sympathetic stimulation on the heart increases the force of contraction of the heart. Okay, right. So let me just go into this in more detail, okay, because this is going to be important in a moment that we understand one of the important proteins in this, specifically phospholamban, we want to understand. Okay, right, so we know that the most important adrenoreceptor on the surface of the cardiomyocytes is the beta-1 adrenoreceptor. So I'll draw in a beta-1 adrenoreceptor here. Okay, so here is our G protein coupled receptor, and this is representing the beta-1 adrenoreceptor, which I'll abbreviate down to AR. Okay, so I might even colour it in specifically. Right, so what's going to happen then is we've got this beta-1 adrenoreceptor on the surface of our cardiomyocyte. This is going to get its ligand bound to it, which will either be noradrenaline or it will be adrenaline, which I'll just abbreviate down to A. Okay, so one or the other is going to bind to this beta-1 adrenoreceptor on the surface of our cardiomyocyte. And of course you'll have loads of these all over the cardiomyocyte, and they're all going to be stimulated when you douse the cardiomyocyte in adrenaline or noradrenaline. Okay, that's going to activate the beta-1 adrenoreceptor. It will change conformation, and now it is capable of activating GS heterotrimeric G proteins. So let me draw a GS heterotrimeric G protein here. Okay, so here's the alpha subunit. This is alpha S. Now, initially it's going to be in the off state, so it's going to have GDP bound to it. Okay, and when it's in the off state, it's capable of binding to another little complex of proteins, which contains a beta subunit and a gamma subunit. Now, we don't care what the specific beta subunit that's going to be here or what the specific gamma subunit that's going to be here is. Okay, it doesn't matter. Okay, it seems as though it varies all over the place, basically. So different alpha S subunits will have different beta and gamma subunits bound, and it doesn't seem to affect the function of the heterotrimeric G protein which beta or which gamma subunit you have bound there. Okay, right. So the beta and the gamma subunit are separate proteins. However, they always remain bound to each other. Okay, and they are therefore often referred to collectively as the beta-gamma complex of the heterotrimeric G protein. So the entire thing here is known as the GS heterotrimeric G protein. So let's colour in the different portions here. So we have our alpha S subunit here. Okay, which is the special subunit, because this can be in two states. This is the subunit that can be in an on state and an off state. Okay, so in the on state, the alpha S subunit will have GTP bound to it, guanosine triphosphate, and in the off state, it will have guanosine diphosphate, GDP bound to it. Now, in the off state, when it's got GDP bound to it, as it currently is, it will then bind to a beta-gamma complex here. So let's colour in the beta-gamma complex in blue and green. So here is a beta subunit in blue, and here is the gamma subunit in green. The other important thing to say about heterotrimeric G proteins, okay, is that the subunits of the heterotrimeric G protein are attached to the inner leaflet of the phospholipid bilayer. Okay, so they're floating around underneath the cell membrane, and the way that they are attached to the cell membrane is that they have lipid moieties attached to them, which implant into the inner leaflet of the phospholipid bilayer and then hold them at the inner leaflet of the phospholipid bilayer. Okay, so basically the idea is here that you attach onto the side of the protein a huge great lipid molecule, generally a very, very long chain hydrophobic structure. Okay, and this is not going to be very soluble at all. Okay, so what instead happens is this lipid moiety, which just means a group of some sort, sticks itself into the inner leaflet of the phospholipid bilayer, this inner layer of phospholipids, okay? And that therefore holds the protein dangling underneath the phospholipid bilayer, okay? So this is known as the inner leaflet of the phospholipid bilayer. Okay, so the alpha subunit of the heterotrimeric G protein and the gamma subunits of heterotrimeric G proteins both have lipid moieties attached to them. It's different lipid moieties between the two. Okay, so alpha S has a very different lipid moiety to gamma. Okay, but the principle is all we need to know here. We don't need to know the specific lipid moieties. The principle of attaching on the lipid moiety so that it will attach you to the underside of the phospholipid bilayer is what's important. Okay, right. So the entire thing then is called the heterotrimeric GSG protein, okay, named after the alpha subunit. You don't care what the beta or the gamma subunits are, okay. GS means that the alpha subunit is specifically alpha S. Okay, right. So when the beta-1 adrenoreceptor becomes activated by noradrenaline or adrenaline, what's going to happen is it's going to change conformations so that it can now interact with the heterotrimeric GSG protein, and what it's going to do is it's going to catalyze the removal of the GDP from the alpha S subunit and the addition of a guanosine triphosphate molecule into that binding site there on the alpha S subunit. So basically it catalyzes the transformation of the alpha S subunit of the heterotrimeric G protein from being in the off state to being in the on state. Now when the alpha S has GTP bound to it, it no longer wants to associate with the beta and the gamma subunits, okay? So they cleave apart and they go off on their separate adventures. Okay, so here now is alpha S here, still attached to the inner leaflet of the phospholipid bilayer, but it's now in the on state. It now has GTP bound to it, and therefore it's no longer associated with the beta and the gamma subunits. Okay, then what's going to happen is it's going to go off and interact with an adenyl cyclase enzyme. Now, adenyl cyclase enzymes are integral membrane proteins. Okay, so let me show you the structure of an adenyl cyclase enzyme. I will put this here. Okay, so they have a rather fancy structure. They have two clusters of 6 membrane-spanning alpha helices. Okay, there's the first one. Here is the second one here. Okay, like so. So this is the structure. So here's the amino terminus of this adenyl cyclase enzyme. Here's the carboxyl terminus. Okay, so as I said, they have two clusters of 6 membrane-spanning alpha helices. So here is this first cluster. And this first cluster of 6 membrane-spanning alpha helices is known as TMD1, which stands for transmembrane domain. Okay, so the T is for trans, the M is for membrane. Okay, then the D is for domain, so transmembrane domain 1. And then the second cluster of 6 membrane-spanning alpha helices over here is then called transmembrane domain 2. Okay, so this is known as TMD2. Now, even though the transmembrane domains look mightily impressive, okay, they actually just hold it at the cell membrane. Okay, they are not the actual active site of these enzymes. The active site of the adenyl cyclase enzyme is going to be made up from part of this loop between transmembrane domain 1 and transmembrane domain 2, and this loop between the two TMDs has a special name. It's known as the C1 loop. Okay, and the C1 loop can be split into two separate domains, C1a, the first portion, and then C1b, the second portion. So let's colour code it up. C1a is here in orange, okay, C1b is here in green, okay. And in fact, that's not all, and the active site is also made up by a portion of this portion that's after TMD2, okay, and the portion after TMD2, so this portion of the polypeptide here is known as C2 domain, and C2, again, like C1, can be divided into two separate portions, C2a and C2b, okay. So I'll colour in C2a here in orange, and I'll colour in C2b here in turquoise. Okay, right. So there's a reason that I have coloured both C1a and C2a in orange, and it is because C1a with C2a forms the active adenyl cyclase enzyme which produces cyclic AMP. Okay, so in order actually to activate the adenyl cyclase enzyme here, what you need to happen is you need the C1a domain to dimerize with the C2a domain. You need those two things to join together, okay, and then they will form an active enzyme which will convert ATP into cyclic AMP and pyrophosphate. Okay, so this picture that I have drawn you here is the inactive adenyl cyclase enzyme. basically the way that alpha S GTP subunits, so the on-state alpha S subunits, activate adenyl cyclase enzymes is by binding to C1, okay, and binding to C2, and bringing these two domains close together to potentiate the possibility that C1a will dimerize with C2a. Okay, so it promotes C1a, C2a dimerization by bringing the C1 domain and the C2 domain close together and getting them to talk, basically. Okay, and that's how it promotes the activation of the adenyl cyclase enzymes, so the dimerization of C1a and C2a. Okay, and then what will happen is, once the adenyl cyclase is active, it's going to take in adenosine triphosphate, ATP, convert it into cyclic AMP, CAMP, and also the other little product you'll get off is pyrophosphate, PPI. Okay, right, and the cyclic AMP is going to go into the cytoplasm. The cyclic AMP is then going to activate protein kinase A. Okay, now protein kinase A is not just a single protein. Protein kinase A is actually what's known as a hollow enzyme. Okay, which means that it's actually made up of multiple proteins stuck together. Okay, so let me just show you the protein kinase A hollow enzyme. Okay, at least the inactive form of protein kinase A is a hollow enzyme. The active form of it isn't a hollow enzyme, it's an individual subunit of the hollow enzyme. Okay, but we'll see that in a moment. Okay, so the inactive hollow enzyme looks like so then. Okay, or at least in my cartoon is going to look like so. So it consists of 4 separate proteins stuck together. Okay, now the first two proteins that I'm going to show here are what are known as the regulatory subunits of protein kinase A, and these do not actually catalyze the phosphorylation of proteins. So these two subunits that I have shown here in these bizarre shapes, these are what are known as the regulatory subunits of protein kinase A. So this arrow specifically points to a regulatory subunit of protein kinase A. Okay, so in red here, this is a regulatory subunit of protein kinase A. And then we've got another one here. Okay, another identical one here. Like so. And those little invaginations that I've drawn here, these are binding sites for cyclic AMP. Currently they are unoccupied. Okay? Now, the other two components then of the protein kinase A hollow enzyme are the two catalytic subunits of protein kinase A, sitting here and here. So this is a catalytic subunit of protein kinase A. Okay, right. So, at present, when you have the inactive hollow enzyme of protein kinase A, which is exactly what I've drawn you here now, 2 regulatory subunits of protein kinase A bound together with 2 catalytic subunits bound to that. The catalytic subunits, which are the portions which are actually capable of catalyzing protein phosphorylation, they are kept inactive by being bound to the regulatory subunits. Okay, so whilst they're bound to these regulatory subunits, they cannot do anything. Okay, so what's going to happen is when cyclic AMP goes up in the cytoplasm of the cell, cyclic AMP molecules are going to bind in these 4 cyclic AMP binding sites, and that causes a change in conformation in the regulatory subunits of protein kinase A, which I'm going to show very bluntly by them going from being in that sort of L shape to being in a straight shape, like so. Okay, so here is this vast conformational change in the regulatory subunits of protein kinase A upon binding cyclic AMP. So I suppose I should probably actually draw some cyclic AMP molecules bound in the cyclic AMP binding sites. Okay, I'll just move this up. So we will represent cyclic AMP as little blobs. Okay, so cyclic AMP is being represented as a little blob, which can be in turquoise. Okay, so you now end up with these 4 cyclic AMP molecules bound to the regulatory subunit dimer here, okay, 2 to each regulatory subunit, okay, and then what happens is the regulatory subunits, once they've bound cyclic AMP, they release the catalytic subunits of protein kinase A, and the catalytic subunits of protein kinase A then go off and phosphorylate proteins, basically. Okay, right. So that's all lovely, but we haven't actually seen how this is going to increase the force of contraction of the cardiomyocytes. And for that we need to know, what do the catalytic subunits of protein kinase A phosphorylate? Well, they phosphorylate a protein known as phospholamban. Okay, and this is a key player. This is present in the sarcoplasmic reticulum membrane. Okay, so this membrane here now, another phospholipid bilayer, is now the sarcoplasmic reticulum membrane. So this is the SR membrane. Okay, so what's going to happen is the catalytic subunits of protein kinase A are now going to come and phosphorylate a protein that is within the membrane of the sarcoplasmic reticulum here. Okay, and for short, this is abbreviated as PLN, and this stands for phospholamban. Okay, so let me colour in phospholamban in a specific colour. So we'll have phospholamban represented in vivid purple. Now, before I tell you about what phospholamban being phosphorylated is going to do, I need to tell you about what phospholamban's normal physiological function is. Okay, so phospholamban normally binds to circa 2A. So let me put circa 2A in here. Okay, so remember the sarco/endoplasmic reticulum calcium ATPase 2A. Okay, now you have a huge number of circa 2As within your sarcoplasmic reticulum membrane. Okay, a certain fraction of them will be free and on their own and not bound with a phospholamban. But some of them will be bound to phospholamban, and those that are bound to phospholamban are not currently functional. Okay, so when circa 2A is bound to phospholamban, it's not functional. So some of the circa 2As that are in the cytoplasm, in the sarcoplasmic reticulum membrane within the cardiomyocytes, are going to not be active because they have phospholamban attached to them. Okay, now you can probably guess what's going to happen. The catalytic subunits are going to phosphorylate the phospholamban, okay, like so, and when phospholamban gets phosphorylated, it then detaches from the circa 2A. So these extra circa 2As that previously were not active because they had phospholamban added onto them, these are now going to be activated. Okay, so you're going to increase the amount of circa 2As within the sarcoplasmic reticulum membrane that are actually active. What is that going to do? Well, this is why understanding the normal calcium homeostasis was so important. Okay, so let me get the picture back again. Okay, remember, there was this equilibrium between the amount of calcium being released by the leak and the amount of calcium being pumped in at any time by circa 2A. If we have now just increased the number of circa 2A pumps that are actually active in our sarcoplasmic reticulum membrane, then we are going to increase the amount of calcium coming into the SR. Okay? If we're going to have an equilibrium, that means that the calcium leak has to go up to match the amount that we're now bringing in. Okay, how can you increase the calcium leak? Only by increasing the concentration of calcium in the sarcoplasmic reticulum lumen, rather. Okay, if you increase the concentration of calcium in the lumen, then of course the leak is going to increase as well. Okay, just because everything is always dependent on concentration. Okay, so basically, once you've increased the number of circa 2As in the sarcoplasmic reticulum membrane, the only way you can be at equilibrium now is for the calcium concentration to be higher in the sarcoplasmic reticulum lumen. Okay, quite simply, if you can imagine this, if you initially, let's just say the protein kinases have just phosphorylated the phospholambans, that's released a whole bunch of new circa 2As to now be active. That's going to mean now that the amount of calcium that comes in is going to be greater than the amount being leaked out. So now what's going to happen is calcium is going to build up in the lumen of the sarcoplasmic reticulum until it is high enough that the amount that leaks out is now great enough to balance the amount that you're bringing in with this increased circa 2A activity. So basically what you're going to do is increase the amount of calcium stored in the sarcoplasmic reticulum at equilibrium. Okay, so by activating this increased reserve pool of circa 2As, you increase the amount of calcium stored in the sarcoplasmic reticulum. Now you might say, well, this calcium had to be taken from the cytoplasm, so surely now the cytoplasmic calcium concentration is going to fall. But of course that will just be taken into account by the sarcolemma, okay, because remember the sarcolemma is only going to be in equilibrium when the cytoplasmic calcium concentration is 10 nanomolar, okay. So to put this another way, yes, temporarily the calcium concentration might fall a little bit in the cytoplasm, but then if that does fall, then the amount of calcium that is being extruded will go down, because of course if the calcium concentration in the cytoplasm has gone down, that means that the concentration of the substrate for these mechanisms has been reduced. Okay, so you're going to release less out of the cytoplasm into the extracellular fluid, whilst the leak in will remain the same. Okay, so again that's going to distort the equilibrium, and it will result in more calcium being brought in because the leak will now beat the amount being extruded, and then that will bring the cytoplasmic calcium back up. So because of the way this is all equilibriumated, everything just fixes itself basically. Okay, so by increasing the number of functional circuitries then in the sarcoplasmic reticulum membrane, you increase the amount of calcium stored in the SR. That means that every time the cardiomyocyte now contracts, every time you now get calcium spikes, there's more calcium in the sarcoplasmic reticulum to be released, and that results in the amount of calcium being released from the type 2 ranitidine receptors going up. Okay, so all the calcium sparks become bigger, and that is what causes the calcium spike, which is the combined effect of all the calcium sparks to be increased. And then when you have that bigger calcium spike, you then get a greater force of contraction generated by the cardiomyocyte. Okay, right. So that now finishes our discussion of normal cardiomyocyte physiology. In the next video what we'll do is we'll discuss what has happened to these calcium signaling mechanisms in the hypertrophied cardiomyocytes.